Welcome back to another video, people. Today we are going to be talking about the polymerase chain reaction, PCRO for short. Now, what is PCRO? The polymerase chain reaction is one of the most revolutionary techniques in molecular biology that has been invented for the replication and amplification of DNA. This technology was invented in the year 1983 by Mullis, who also won a Nobel Prize for his invention in 1993. Now, what Mullis sought to do was to improve on an existing technique of DNA replication. Now, years ago, DNA amplification was done using biological and biochemical methods that relied on living cells such as bacterial cells. Now, what would happen years ago was that they used recombinant DNA technology, wherein a plasmid was taken from a bacterial cell and the gene of interest was taken from the human cell. Now, that gene of interest and plasmid were both cut using something known as restriction endonuclease, after which they were joined by using something called DNA ligase and then inserted back into the bacterial cell, after which this bacterial cell was then cultured and obviously multiplied. So all of this is known as recombinant DNA technology, and I will be covering that in another video. But basically, this is how genes were amplified before the invention of PCRO, and this process would take weeks to accomplish, because after multiplying this bacteria in culture, you'd have to extract the DNA again. Now let's go into the concept and principle of PCRO. But to understand how PCRO works, you must first understand how genes and DNA are amplified in living systems. To understand this concept, we need to look into the central dogma of molecular biology. Specifically, we'll be looking at replication in a little detail. Now, you can see that you have a double-stranded DNA that is first of all acted on by an enzyme known as helicase, which splits or unwinds the double helix into two single-stranded DNA. And you have free-floating nucleotides. These are deoxyribonucleotides, your adenine, thymine, cytosine, and guanine, just floating about. Now you have another enzyme known as DNA polymerase that comes and starts to add these free-floating nucleotides to the single strand, making a new double strand. So there's DNA polymerase down here, there's another DNA polymerase up here. So you have this your double-stranded DNA, now two strands, two double-stranded DNA. Okay, so this is basically what happens in the cells of living organisms. Now, PCRO is an in vitro DNA replication, so it's an in vitro version of what happens naturally. But instead of using the enzyme helicase, it uses temperature cycles to separate DNA strands. So an increase in temperature separates DNA strand rather than an enzyme. And thereafter, other processes follow that allow the amplification of this single DNA strand. And this amplification is exponential. So the first cycle, you get two identical DNA strands, and thereafter 4, and then from 4, 8. We are going to be looking at the process in a little more detail. Now we're going to look at the components of PCRO that are used to achieve the amplification we've discussed. Now first, you need a template DNA, which is the DNA that you want to replicate or amplify. You need Taq polymerase. As we have seen in the natural replication process, there is a polymerase enzyme that allows for the binding of the free nucleotides on the single strand. But Taq polymerase is a special kind of polymerase extracted from a microorganism known as Thermus aquaticus. This microorganism exists in hot springs, so it is quite heat resistant. You also need free nucleotides that are going to be bound and attached by Taq polymerase. You also need primers. A primer is a short single-stranded oligonucleotide that defines where the start and end of the region to be amplified. You also need magnesium chloride and PCR buffer. These help to maintain enzyme activity and reaction conditions. Now, all of these components are put into a PCR tube and thereafter put into a thermocycler for the reaction process. The PCR tube is placed into the thermocycler where the reaction takes place. Now, the target DNA undergoes 3 steps per reaction cycle. First is the denaturation step, where the temperature is increased to 95 degrees and the double-stranded DNA is denatured and broken into 2 single-stranded DNA. Thereafter, you have the annealing phase, wherein the primers attach to the ends of the target DNA. And then in the extension phase at 72 degrees, Taq polymerase binds to the primers and inserts the free nucleotides to the growing chain from 5' to 3' of your target DNA. Now the same trend continues into the second reaction cycle, this time yielding 4 double-stranded DNA. The third reaction cycle would yield 8 double-stranded DNA. Now, a typical PCR runs for about 25 to 35 cycles, taking about 2 to 3 hours, and this would yield billions of identical DNA. Now, the thermocycler is the machine used to carry out the PCR reaction. It automates the temperature shift from heating to cooling the sample precisely and ensures reproducibility and the yielding of billions of copies of high fidelity. So this is what the machine looks like. You can see that this is the sample chamber. There is a heating block within this unit where the sample vials are kept that allows it to get hot. And this is the digital display where you can see the different temperature shift per cycle, and all of that can be adjusted using these buttons. So this is what the machine looks like, and there are newer models that have this drawer type design. Now we go to some applications of this technology. Now in virology, the PCR has been used in the detection and genotyping of viral nucleic acids. So it can be used in the detection of diseases like HIV, COVID-19, and so on. In microbiology, PCR has been used in the rapid identification of pathogens. In forensic science, it has been applied in DNA fingerprinting and identification from trace samples of DNA left in crime scenes. This has led to the arrest of criminals. It has also been used in genetic diagnosis for detecting mutations, deletions, or insertions in disease genes. PCR is also used in cancer research for detecting oncogene mutations like RAS or other kind of mutations in cancer cells, such as that in P53 and so on and so forth. Now, in summary, PCR is a rapid, powerful method of DNA amplification based on repeated cycles of denaturation, annealing, and extension. And this tool has revolutionized molecular biology, medicine, and forensic science in a number of ways. So this is the end of this mini lecture. I hope you gained a thing or two. Please remember to give this video a thumbs up, subscribe to the channel if you haven't already, and let me know in the comment section if you have any questions or need clarity on any of the concepts covered. I will see you guys next time. Bye.